Okay. Well, we might just get started. Once Amy has arrived, maybe we can go back and do the quick intros. But I really just wanted to say a big welcome to everybody. We're really excited to collaborate with the BDSRA Foundation and BDSRA Australia. Together, we came up with the idea of a research year in review because we've had so many interesting discoveries and updates throughout the year. Some not so great, but we'll talk a little bit about those too. But really, this is just a way for each of us to go around. I'll ask the panelists to each introduce themselves briefly. And we've together worked as a group to really pull out, I guess, cherry pick the highlights for each of us throughout the research year in 2023. And I'll be introducing those topics, but then handing over to each of our panelists to discuss a little more. I can see, okay, oh, here we go. Here's Amber. Okay, Amber, do you want to just give a quick introduction?
Yes. Hi, good morning, everyone. Sorry, I've come in through a different link, so I can't actually see myself. So am I talking to the camera or am I talking?
We can't see, your camera's not on, so.
Okay, I can't, with the access that I've got, it's not allowing me to put my camera on. I'm sorry.
Oh, okay.
It's, yeah, I've had to come in through the registration link, not the panel link that you sent through. That
No
didn't
problem.
work. So I
Sorry,
apologize, everyone. You can't see me. I'll try and figure it out while the meeting's on. But good morning from Australia, and I think it's good evening or good afternoon, depending on what corner of the globe you're coming in from. Yes, my name's Amber Rundle. I'm the president here for the patient group organisation in Australia, which is obviously Batten's Disease Support and Research Association Australia. I've been the president just for the last year, but I've been on the committee for quite some time. Basically, our main mission here in Australia is not dissimilar to the BDSRA Foundation in the US. We're here to support our families, so we do a lot of work in the family support area. We do a lot of work in raising awareness and obviously in advocacy as well. And then our big other mission item, I would have to say, is our very successful research grants program here in Australia, which we're very proud of. In the last 4 years, we've managed to put 400,000 Australian dollars towards incredible research, and we're continuing to do that. And so we're just a small group here in Australia. We also look after New Zealand as well. They're kind of within our reach. But yes, thank you to everyone here this morning, our fabulous panel. We're looking forward to the update, and enough from me. Thank you all for coming. I'll hand back over to Ineka.
Thank you. And Amy should be joining us very soon from, I think, flight delays heading to Washington today. But let me pass around. Now is a good time to introduce our other panelists. So I'm just going around as you appear on my screen. So Professor Tony Cook.
Hi, Ineka. Thanks, everybody, for coming along today. I'm Tony. I'm all the way down here in Hobart at the very southern end of Australia. I work predominantly with stem cell models of disease and have had a focus on CLN3 disease over the last little while. Yeah.
Okay. Professor John Cooper.
Hi, I'm John Cooper. I'm at WashU in St. Louis, and I've been working in the Batten field for quite a long time. We're interested in figuring out where problems are happening, both in the brain and in the rest of the body, and then trying to intervene to stop it. I organized the previous one of the NCL meetings in St. Louis a couple of years back, and you probably know me. I pop up at the BDSRA, and at Ineka's prompting, we reorganized the science session, and I hope that was useful for you.
Dr. Courtney Santucci.
Hi, everyone. I'm Courtney Santucci. I'm a pediatrician in Denver, Colorado. So I'm at the Children's Hospital of Colorado, where we have a neurodegenerative multidisciplinary clinic where I have the great pleasure of caring for patients who have Batten disease. I'm very happy to speak with y'all today.
Thank you. And last but by no means least, Dr. David Pierce.
Dave Pearce, longtime Batten disease researcher and current board member for the BDSRA.
Thank you. All right, so moving on to our topics for today. So we've sort of grouped these highlights according to the understanding the basic science, so understanding the genes and proteins responsible for the genes for the disease, particularly CLN3 and CLN5 have had some major discoveries this year. We'll look at some clinical observations and new understandings of clinical presentation of CLN3 disease, and some emerging therapies as well. In CLN2 disease, we'll look at some learnings from our existing therapy, which is the Brineura or solipinase alpha, and some of this real world data and understanding of it, of how it works and over the longer term in our children. And emerging therapies, excuse the typo, we'll have a look at emerging therapies in CLN2 disease as well. CLN5, 6, and 7 have also had some interesting discoveries this year in terms of the animal models of the diseases, and one clinical study, a gene therapy as well. So most of these findings were presented at some point throughout the Congress in Hamburg. It was a wonderful 5 days. All of us on the panel here today were in attendance. And it was just, yeah, I guess a really great whistle-stop tour of all of the work going on around the world, and the highlights and major findings from those groups. So we've cherry-picked some of those best findings for you today. John, I'll pass over to you for a little bit of background here.
Okay, so one of the things we want to do is figure out what's going wrong in these diseases. And the first step is to try and understand what the proteins that are missing in this disease because of the mutations, what they would normally do. Now, to be perfectly honest, we don't really know what that is, but we know it must be really important, because when it's not there, we get a devastating disease. And we think in part, this is due to how things move around inside the cells is really important. And there is now evidence, Ineka, that CLN3 may be involved in how things move around inside cells. There's also a lot of excitement. Some big news emerged about what is building up inside the lysosome, that waste disposal unit in CLN3, and it's a very particular type of fat or lipid. And we think that gives us important clues about what CLN3 might actually be doing. And it also gives us a biomarker for measuring potentially whether a treatment has worked. And lastly, CLN5 has turned out to be very interesting. Most of the enzymes, like CLN1 and CLN2 enzymes, break things down. And when they don't work properly, stuff builds up. Well, it turns out that CLN5 does almost the complete opposite, that it actually makes something. It makes a particular type of fat or lipid called BMP. And when that process doesn't happen, that causes lots of things to go wrong inside the cell.
I'll pass over to Tony now, our resident CLN3 expert.
Thanks, Ineka. So, as John mentioned, we don't really know what CLN3 does. But what we do know is that it's involved in very important things to do with lysosomes. But how CLN3 gets there is we have our DNA in our nucleus of our cells that's converted into an mRNA copy, and then a peptide and a protein. And one of the new discoveries that we found about CLN3 this year came from Sarah Moll's group at UCL in London, and that there's actually quite a large number of different CLN3 mRNA forms. And about 1/3 of these encode for a non-standard or slightly different CLN3 protein. And furthermore, there's one of these forms seems to be quite enriched in the brain. So this is new data. We don't quite understand what this means, but it adds to the complexity of our task in trying to understand this disease better. And so we need to conduct functional studies on these various forms of CLN3 to try and work out which ones are the important ones so far as the disease is concerned. Once the protein is made and it becomes embedded in the membrane of the lysosome, it performs a particular function there. And that's what we don't know, but we're starting to tease that apart. And Hannah Best in Cardiff presented some data showing that CLN3 pumps a particular ion, so potassium, indicated by this K symbol here, out of the lysosomes. And so when this is defective or not occurring in CLN3 disease, that alters the ionic environment within the lysosome. And that can change the function or the activity of the other enzymes in the lysosome. What we don't yet know if CLN3 is involved in the transport of other molecules yet. And so the next slide, Ineka, please. Thank you. So John mentioned that things move around the cell, and that's important for their function. And one of the more exciting things that we learned this year about CLN3 is that it seems to be quite involved in this process. And so this first paper on the right-hand side here talks about CLN3 being involved in trafficking of this protein M6PR. So this is a really important protein that actually is involved in guiding a lot of lysosomal enzymes into the lysosome. So the fact that this CLN3 interacts with this protein kind of puts it at a bit of a hub for a lot of lysosomal enzyme trafficking, and hence that might be part of the reason why we see such a striking phenotype in CLN3 disease.
So moving on, the Weimar lab is presented and has a paper out there. It talks about a protein called sortilin. So this is another one of the nuts and bolts in the cell. And if you sort of think about the name sortilin, this is it, what Tony was saying and what John is saying about, moving things around the cell. Imagine if you think of all of the NCLs as balls, right? You're trying to juggle them all, and where do they need to go if you need to place them in a particular place? So sortilin is another piece of the puzzle, which is involved in potentially placing all of the different NCLs or the CLN proteins or nuts and bolts around in different places. And as we've heard, some of these can be in different parts of the cells. So if we actually transition, and I'll mix the sortilin with the CLN5 BMT story here, is many of the CLNs actually reside in membranes. Membranes are basically pieces of fat. Because what they are very good at is housing the all sorts of proteins, but in particular relevance here is the CLN proteins. So they sit there and allow things to go across the membrane, so in and out of certain compartments within the cells. So what the CLN5 gene has been identified to code for something that actually makes this one particular type of precursor or building block for certain types of fats, which are very important for the construction and really the assembly of membranes. So one of the paradoxes of Batten disease is we always hear about the lysosomal storage material in similarities, and we've never been able to map the fact that these proteins actually are in the same pathway because they don't physically interact with each other. But if you have defects, they all lead to the similar sorts of pathology that John tells us about all the time. So we're leaning in the direction now where is some of the metabolism, so maybe fat metabolism or fat assembly in the membranes, maybe that intersection and being the relationship as to why we see some of these similarities. And this is actually an important finding. The last time that a CLN actually had a function identified, I don't know, John and I were wee nippers in the NCL field when they identified what CLN1 and CLN3s and CLN2 were doing. So this is really exciting and is going to help us understand all of the NCLs, I believe.
Yeah, my hair wasn't grey then.
Or so long. In fact, I had a look at that CLN5 paper just quickly this morning to get this screenshot, and it's had thousands of sightings and downloads already. So just to give you a feel for the impact that this discovery has had on the wider biology field as well, cell biology field, not just in the NCL field. Our next topic is around the clinical observations and our understandings of the different symptom load on not just CLN3, but other subtypes as well. I'm going to pass over to John at this point.
This comes around because we spend a lot of time trying to understand the brain and trying to treat it. And when we do, it doesn't completely treat the disease in our animal models. And that got us thinking of disease outside the brain. And the other main reason was we listened to a lot of families telling us about disease outside the brain. So one of the things we look at are the nerves that come from the central nervous system that help us feel things. sense things and help us move around. And what we've shown is that our mice are more sensitive initially to pain and then become less sensitive. There are problems with the nerves that innervate the muscle. We think this is part of the muscle wasting itself and underlies some of the problems those mice have with walking. And we think there are actually problems in the muscles themselves. And the good news is that we can actually improve this by putting gene therapy into the bloodstream of both CLN1 and CLN3 mice. Now, the other main area we're really interested in is bowel problems, and many families told me there were those problems. And I figured that this might be because the nerve cells which die in the brain, nerve cells somewhere else might also die. And you have 500 million neurons in your bowel in something called the enteric nervous system, which is really important for how things move through the bowel. So what we have shown is that, yes, those neurons do indeed die in the bowel of CLN1, 2, and 3 mice, and the few cells that are left look really sick. That causes things to go slowly through the bowel and to back up. But again, we're trying to treat this, and the good news is that we can, again, using the same gene therapy method, actually prevent that slowing of things moving through the bowel and protect many of the neurons. And we've done that successfully in CLN1, 2, and 3 mice, and it just emphasizes that we need to think outside the brain when we're thinking of trying to treat the whole disease. And again, this was all prompted by parents talking to me.
Another learning that we've had in the CLN3 disease space from the clinical sense, Courtney's going to talk about now.
Yeah, I think this was really interesting to me to hear about in Germany, because it's definitely something that we've observed in our patients with CLN3, particularly as they get into their later teens. So the basic topic here is that some have published about how the parasympathetic function of the nervous system diminishes and kind of is lost as the disease progresses. Basically, what your parasympathetic tone or function of your nervous system should be doing for you is kind of all of your rest and digest functions, sort of the opposite of the fight, flight, or freeze mode of your nervous system. Neither of these is good all of the time. I mean, they're sort of meant to coexist happily, like a yin and a yang. But when the parasympathetic tone starts to drop out, what's observed is that we can see episodes of almost like way too much sympathetic tone, or what's known as storming of the nervous system. Symptoms of this include fast heart rates, so tachycardia, flushing of the skin, high blood pressure, hypertension, discomfort, agitation, frankly, fearfulness, sort of a fearful appearance. This study that was presented in Germany looked at 9 patients, all over the age of 15, who were being followed. They noted that the instances of episodes did resolve on their own, usually within an hour. But the interesting thing to me was that they noted that there was always, so they had the children wearing, or the teens, I should say, had them wearing some devices to keep track of their heart rate. And they noted that always there was a preceding heart rate elevation before any of the other clinically obvious symptoms would start. And so it sort of begged the question, could we interrupt these episodes if we could interrupt that initial tachycardia? I think that was really intriguing. Could we palliate this aspect of disease progression, perhaps by trying to enhance parasympathetic tone as best we can? There are some peripheral therapies that can maybe do that, and or regulate the tachycardia, perhaps with something like pacemaker introduction. So I thought it was just really, really intriguing, and something that definitely needs further looking. And just to speak a bit more about some of the preceding papers, some of the earlier papers that have already been published in 2018 and 2023, I think by Dr. Ostergaard, getting at the same witnessed phenomenons of these paroxysmal sympathetic events.
There's a question in the chat box, Courtney, that's relevant, and we will come back and address the ones that haven't been answered just yet. But since this is so relevant to exactly what you're talking about now, someone has asked whether this phenomenon might contribute to the regulation of body temperature as well, which is what we see in those CLN3 kids or adolescents?
Yeah, so poor temperature regulation is definitely an aspect of dysautonomia. So whenever the autonomic regulation of the nervous system is kind of going wonky, you can see lots of irregularities, whether it's the heart rate, the blood pressure, the peripheral perfusion, the gut motility, or the temperature of the body can all just be erratic. So yeah, I think those two things are linked.
Okay, moving to emerging therapies for CLN3 disease. Dave was going to speak to tamoxifen.
Yeah, tamoxifen. So tamoxifen is a drug that many of you may have heard was a first line, or still is a first line of therapy for some types of breast cancer. It's used less and less now as we've understood that the genesis of that much more. So this targets a specific type of membrane protein that regulates really in women hormonal levels of with respect to cell signaling, but also is expressed in pretty much everybody. The idea here is that receptor intersects with what I was talking about, all the balls that juggle. Now, we don't even know exactly how tamoxifen works, but it obviously affects some of that, but that juggling of the balls around the cells. So some folks actually took a look at this with respect to really sort of nullifying or normalizing certain attributes of the balls juggling around the cells. And in the CLN5, they showed in the mice that it seemed to ameliorate some of the disease phenotypes. Certainly in the cells, it was clearing some of the storage materials and some of the pathology. They also looked at CLN3, but they didn't look at the mice, or didn't report in the mice. They just looked in the cells and showed that there was some, again, reversing of the pathology. The CLN3 mice, I just have to add that caveat, are inherently very difficult for us to study in terms of showing severe disease and things that we can actually measure with drugs. So that's not a criticism on that work. This work is relatively old, really, in the field of NCL, a couple of years now. Nothing has really been advanced beyond, understanding that it may have some sort of ability to clear some of the storage material. So, John, I don't know if you've seen anything in addition to that.
No, nothing yet. So, but yeah, work is going on in the background with these things.
And
always, all these findings are being pursued constantly.
Yeah. And moving over to ASOs.
Yeah, thanks, Ineka. So ASOs are a little bit different to other drugs. So down the left side of this slide here, we have a protein that's made from our DNA. And lots of drugs actually work by inhibiting the protein. Some of them activate proteins, but it's much harder to do that with a drug. What ASOs do is they actually interfere at the RNA level in this process from DNA through to the protein. And there's two consequences that an ASO can have. One can be to actually prevent production of the protein. The other is to alter the protein in a specific way. And this is the process that the Hastings group has done in developing a CLN3 targeting ASO. And if I can have the next slide, please, Ineka. So this is some work that they published a few years ago. And what they did here is to use an antisense oligonucleotide that induces what we call skipping. And this has been designed to overcome the problem of the 1 kilobase common deletion that we see within the CLN3 gene. And so that's kind of diagrammatically depicted on the bottom left-hand corner of this slide. So what they presented at in Hamburg was a new pig model where they've engineered this pig to have this same 1 kilobase deletion. And they've designed a pig-specific ASO that does the same thing as what they showed happens in mice and happens in human cells. They've shown that it's able to induce the exon skipping, and they have studies underway looking at if it's able to modify the disease phenotype in these pigs. But the implications for this sort of therapy are that it's going to be specific for that genotype. So it's probably only going to be useful for people with a homozygous 1 kilobase deletion. And so that leaves still an unmet need for people with other genotypes, or even if they have one copy of the deletion. And as I mentioned, it's harder to restore protein function than it is to inhibit it. And so what they're trying to do here is create a scenario where they're restoring some of the function of the CLN3 protein. That's going to be difficult. So the dosing of the ASO will be very important, and it may be that this actually slows the disease rather than halts the disease progression.
Moving over now to the inhuman studies in CLN3, and Courtney's going to talk us through the Batten 1 studies.
Yeah, so and I know that there have been separate meetings offered to families to learn about Batten 1 and what comes next. But presented in Hamburg was the results from the phase 1 and 2 trial, at least 1 year in. And in short, presented was a good safety and efficacy. These are very encouraging. And so one of the things that's been sought after, I would say, in looking for disease markers of CLN3 progression has been a biomarker, so something that you could run a lab on, something that circulates in the serum. So what you're looking at here are serum levels of neurofilament light chain demonstrated to be reduced in patients over time, which is a good thing. And I would say, in addition to showing promising effects by that biomarker reduction, just the safety and relatively good tolerability of the miglustat drug were shared about. Clinically, and I think it's early days, and I think it's going to be really important to look at what happens when patients are treated younger than were in this phase 1 and 2 trial. But there was some slowing of the expected worsening of motor symptoms as assessed by the BDSRA scales when these patients were coming back in for their clinic visits, compared to what would have been expected with natural progression of their disease. So all in all, promising for sure, and we're looking forward to now phase 3 beginning. And so this is going to be starting in the coming year. Hopefully any CLN3 parents and families are already aware of this. If not, listen up. The goal here will be to enroll 60 to 70 patients between the ages of 4 and 16. They will be sort of clustered into 3 different age groups, sort of the little ones 4 to 8, the middle-aged ones 9 to 12, and then the older young ones who are 13 to 16. The dosing will be with, next slide, you just advance it, Ineka. So it's an oral medication, it's a liquid, and dosing does have to be 3 times per day, which is a bit of a bummer. So it's not the world's easiest medicine to take. Anything you have to take several times a day can be difficult, but we're hoping that it's going to make a nice difference and be well tolerated. The trial, like I said, will begin enrolling in the early part of this coming year. So right after the new year is what we're expecting. There are going to be 3 sites in the US. They will be in Baltimore, Maryland at the Kennedy Krieger Institute with Dr. Augustine, in Columbus, Ohio at Nationwide with Dr. De Los Reyes, and in Houston at Texas Children's with Dr. Clark. 24-month trial. It is placebo matched, placebo controlled. So there's a chance for those enrolling that they would be getting the placebo and not the active medication. And the aim is that the study would close in 2026, and hopefully if efficacy can be shown, that the drug could be then approved and available for just routine prescribing as early as 2027.
Mm-hmm. Just to supplement that, I can let you know, so as Courtney mentioned, the 3 sites in the US, we also have one in Germany, in Hamburg, Netherlands in Utrecht, Sweden is Gothenburg, UK is London, the Gosh Hospital, Australia, one site at the Sydney Children's, and Denmark at the Aarhus Hospital as well. So, and if for anyone wanting further information on that, the Beyond Batten Disease Foundation did recently host a family webinar with all of those details, more detailed protocols, inclusion, exclusion criteria, and so on. So please reach out to us at the BDSRA. We can connect you as well with that foundation to find out more information if you need, and to connect you with the study sites as well, because we know there's some internationals on here as well. Sorry, the endpoints, we're still talking.
Sorry, I forgot what we were going to say about the endpoints. So how will the study assess whether the medicine is helping? The primary endpoint is actually going to be one of determining visual acuity. This will be just by patient being able to count fingers and recognize hand motion a certain distance from their face, and perceive light. All involved recognize that in CLN3 disease, vision loss is an early symptom, and by the time of study enrollment, certainly by the age of 16, we would expect a significant impairment on vision. And so there are other endpoints that will also be tracked, and these include just sort of cognitive function measures and motor function measures. The biomarkers, so the blood test that I mentioned earlier, the NFL, the neurofilament light chain, will also be being measured along with another biomarker, glycosphingolipids. And it is unfortunately an exclusion to enrolling in this study if the child at the point of enrollment cannot perceive light, or if they have heart abnormalities with an abnormal EKG at the point of screening.
Okay, we'll shift gears to the CLN2 existing therapies. Sorry, this is all you, Courtney, at the moment.
I'm tired of listening to you. So yeah, one of the other interesting things I thought that was presented in Germany had to do with studies that the German group in Hamburg have been doing on their patients who are receiving Brineura, so receiving every 2-week infusions with Ceralac alpha intrathecally. They have had the good fortune to be able to get volumetric MRIs of the brain on each of their patients every 6 months. And so they can really compare the pace of cerebral volume loss over time in the patients who are currently being treated, as compared with historic data that come from similar volumetric MRI measurements done on patients who never got Brineura. And so historically, we expect there would be about 12% of the cortical volume lost each year in a CLN2 patient. And on Brineura, what they shared is that they're saying more like 4% loss for the patients who are being treated, which is really exciting. And then noting particularly that there's loss in the visual cortex, also in the somatomotor cortex. But I thought that was interesting because perhaps if we could also treat these very same patients with an intraocular therapy to slow progression there in the eye, perhaps maybe the visual cortex would hold in better. That's just my own postulating there. Cortical thickness for all of us around the world could be a good biomarker for us to look at following in terms of keeping track of disease progression. And I think the German group are doing great work to set the kind of the baseline data there and give us a good data set for reference against. They also shared about just some clinical outcomes for children who've been receiving Brineura. They have found that basically if children are started on Brineura dosing at over age 3 years, they tend to do similarly to children from the natural history study, so to untreated children. Whereas if we can get treatment started earlier than 3 years, we see a much more impactful arrest or slowing of disease progression. And that's good data for us to use, I think, as all of us go forward advocating for early screening, early detection for CLN2 particularly. Let's see, the specific numbers on their treated cohort in terms of brain volume, so it's 2.8% total volume versus 16% in the natural history study, the total brain loss, and then similar numbers in the cerebellum specifically. Yeah, I think.
Thanks, Courtney. So I'm keeping the train moving. I'm seeing.
Thank you.
Sorry. So the gene therapy is now looking at, John's going to talk us a little bit through this and how his mouse models looking at the benefits of gene therapy have also started elucidating some interesting findings in terms of the seizure phenotypes too. So it's a double-pronged discovery, and I think these studies that John's done.
Yeah, so this is all using a mouse switch Dave actually made a number of years ago, and this carries the same mutation that the most common mutation that most CLN2 children have as a particular point mutation. And we were studying them, but unfortunately trying to figure out the staging of disease, but they had a nasty habit of being very sensitive to dropping dead if you make a loud noise near them or even look at them funny. And we tried to find out why that was. And it turned out that the mice are actually having seizures. So we recorded from their brains in much the same way as you could record from a child. And we saw that they were definitely having seizures that tended to be just before they died. And this is because the seizures are really very severe in these mice. And we wanted to find out why. And there's a particular group of cells in the brain whose job is to control or damp down brain activity. They're called interneurons. And it turns out that specific populations of those in a particular place in the brain are especially sensitive in CLN2 mice. And we're doing a lot of experiments trying to prove whether that's the case or not. But while we're doing those, we obviously want to treat them. And this is where we stepped in with gene therapy early in the disease process and discovered that we can actually prevent those seizures. And because the mice are not dying from those seizures, they now live out to at least 1 year of age, which is great. And the whole idea of doing that work we did in conjunction with Regenex Bio was to try and set up having a clinical trial. But unfortunately, as was covered in a webinar recently, for a variety of reasons, that program has now been put on hold.
So we're looking at, as well as the gene therapy for CLN2 disease in the eye, which is the Regenex Bio program that we recently talked about and will again in the following slide. There is also the intravitreal ERT that has had some interesting findings this year. Courtney?
Yeah, this is a really nice thing that this paper just came out just last week, so just in time for this presentation today. But this was a study that shared about 8 children between the ages of 5 and 9 in the UK, so being treated at the Great Ormond Street Hospital in London, where every 8 weeks for somewhere between a year and a year and a half, the children were taken into the hospital, put under anesthesia, and into their eye was injected Brineura, basically. These were also, of course, children who were receiving every 2-week treatments of intraventricular Brineura therapy. The injections are intravitreal. And what they looked at was basically the volume of the area right around the macula in the back of the eye to see whether they could show any slowing of the macular degeneration that's expected in CLN2. And they did show, well, first they showed great safety with this procedure. So they didn't see things like problems with bleeding into the eye because there had been a needle put in it, or high pressure within the eye because medicine had been put in there, or really any infections or things like that. So great safety profile. They did mention one child had a minor kind of subconjunctival hemorrhage, but when they checked him out, he was found to have a low platelet count, and so for subsequent treatments, he got a medicine to help prevent that from recurring, and it was really considered minor and very, very treatable. But anyway, it's really good early information that treating the eye in CLN2 may help to slow macular degeneration, and seems, at least at this point, to be a safe thing to try.
We don't need to spend too much time on this because, as John said, we did have a recent webinar on this, but can also answer more questions in the Q&A if people have those. So yeah, we'll skip across, and the same with the CNS. So there's an investigator-initiated trial But we're hoping, again, just I guess a quick update too, is Amy and myself are still in communication with Regenex and still hot on their heels to make sure that they're pursuing those solutions to continue these programs. So we're hoping to give you more updates on this in the coming weeks. Okay, moving over to CLN5 and 6 disease, some new learnings that we've had in animal models this year, John.
Yeah, so this is where in the development of the enzyme replacement for CLN2 disease, or Brineura, doing studies in dogs as a large animal model were very important for figuring out how to do the trial in children. And this is a series of work from a group in New Zealand, originally from David Palmer, now led by Nadia Mitchell. They happen to have sheep that have a mutation in CLN5. They have another one that has it in CLN6. And they've been studying them for many years together with us. We helped them with much of that work in the early years. And the idea is, can you treat or test a bigger and more complicated brain than there is in a mouse? And they've recently published encouraging data saying that, yes, you can treat the brain with gene therapy for CLN5. And it's important, if you treat the brain, that gene therapy doesn't reach the eye. So they've also been looking at treating the eye as well, which is work that Nadia presented. And if you put the gene therapy in both sites, you get a better outcome. And that is the basis for a trial, which is going ahead, run by Neurogene, which is the company that they did this work together with. And it highlights the importance of using that large animal model, as we are doing ourselves with a CLN1 sheep, which we had to make by CRISPR with some friends at the Roslin Institute in Scotland. And we're going ahead doing gene therapy in those sheep. And we also heard from a company called Spark Therapeutics, who are also doing gene therapy in CLN1 sheep.
We'll come back to that in the Q&A because there's a question around CLN1 as well, John.
Absolutely.
Finally, this is our last finding for our highlight for the presentation. Dave, you wanted to speak to this one?
Yeah, so the CLN7 gene therapy trial was run out of UT Southwestern. So there was a lot of excitement about this one. It's using the conventional viruses that John has described, intrathecal delivery. The report that came out, the small number of children that were treated, unfortunately, while again it reached safety standards, which is what's the primary endpoint or the first thing we want to measure. Unfortunately, there was no sort of regression, well, really slowing or regression of the actual disease with respect to that. As far as we are currently aware, this trial is now discontinued or not advancing. Whether the investigators are going to do any more with respect to that. And when I actually saw the question on CLN1, Ineka, I was just going to add to that. I mean, somebody actually asked them the questions, what happened to the CLN1 trial with Tasha? They never actually got to clinical trial. They have no money. I mean, that was a separate discussion we had on the other webinar recently, is that there's no resources to do these trials, which is very unfortunate. Investments are just not there. So that's why there hasn't been an update, because the company essentially does not have a program. So Ineka, I know if you wanted to add to that.
Yeah, absolutely. I'll stop sharing my screen so we can all see each other now a little bit better. So essentially, I mean, the update has been for the last, well, nearly 2 years. I think it was February 2 years ago that they made the decision to discontinue or to put the trial on hold until further notice for the CLN1 gene therapy with Tasha. We have remained in communication with them for updates in the background all of this time, and they've been open to conversation, but simply haven't had any updates to share with us other than that they are still pursuing other commercial investors and looking outside the box, not just at commercial investors, but ways to progress and move this program forward. At one stage, we talked publicly about it being a small proof of concept study instead of a full-blown clinical trial. Those are the conversations that are continuing, and Tasha have committed to releasing a statement shortly, because we have asked them this exact question. Look, we haven't heard anything. The CLN1 community hasn't heard anything for a while. We're filling the gap for you, but if you could step in and share some communication with us sometime soon, that would be great. So I understand that that is in the process at the moment. So hopefully that's sooner rather than later, but they are still very much, like others that I guess have left and have not remained in touch with us. I have to say, to give them the credit that they have stayed in communication with us. And I can see they're genuinely wanting to move this program forward as well. They haven't stepped away at this point.
So, I mean, at this point, maybe just to emphasize that, there are other research efforts going on for gene therapy in CLN1 disease. And some of those, as I indicated, are in using a larger animal, a sheep, that was made specifically for the purpose of testing therapies. We did work with enzyme replacement in those sheep and showed we could scale it up from mice to sheep. And now we're doing the gene therapy work. That's work funded by the NIH that we're doing using some particular viruses made for us by Beverly Davison. There are also, as we heard at the conference, Spark Therapeutics are working with the same sheep model. It all comes down to having a larger model allows you to test other ways to deliver it, and it may be to treat a bigger brain that a different delivery route might be more appropriate than the method that was used in mice. And that's the sort of thing we're testing. Where is the best place to put it to get it to spread to a much bigger brain? And that work is ongoing. We're doing this as carefully as possible to get answers that will tell us something meaningful. We've put a lot of work into how do you measure disease in a sheep. They're surprisingly clever. You can test them in a maze. You can do MRI on them, much like you do with the child. You can test how they walk. And we're doing all those things. They are moving forwards. I know they never move forwards as quickly as we want to, but please be assured that other efforts are going on in parallel to anything that you've already heard about.
Yeah. Yeah. Another question about the CLN2 Brineura is still being indicated for children 3 and over. This is in the US. So the label still is only for children 3 years and older. But given this data, do we expect to see? So the question is whether I'm misinformed that this has been changed. It hasn't, as far as I'm aware, but it's in discussion based on these new data. Courtney, you might be able to speak to that some more.
I guess I would just say maybe I'm misspeaking here, but I know that we give Brineura to much younger children, children who are only a couple of months old even, and it is payer approved, sort of, covered by insurance and everything. I imagine that is technically off-label use, and yet totally done. I'm not sure if when Brineura will change its labeling or get broader FDA approval, but it doesn't seem to be impacting on our clinical management. We treat patients as soon as they're diagnosed.
Mm. Yeah, and that holds with other countries as well. In fact, in other countries, there is no age minimum requirement in the label. So those labels or the product information is different from one country to another, and the approvals are different from one country to another. But as Courtney said, yeah, we're definitely seeing children in that infancy being treated too. So again, this gives a really solid argument for why we should be, early screening, newborn screening, and so on. A couple of questions around the storming and the dysautonomia in CLN3. One family, a CLN3 family, is asked, is earthing something to consider? Earthing, I wasn't sure.
I'm just not familiar with it.
Yeah, okay. So maybe Sean might want to quickly type something into that answer to clarify further. But someone else has asked, so in terms of CLN1, is this something we see in other Batten forms as well? So this family see a lot of flushing of the skin, increased heart rate, agitation with their 6-year-old CLN1 daughter. It could also be just general discomfort, but she's interested to know if this could be a parasympathetic issue as well.
That's a good question. I mean, as far as I know, there haven't been kind of systematic studies looking at any of the other forms to kind of quantify these sympathetic storms. But it stands to reason that really any degenerative process of the central nervous system could result in dysautonomia. I mean, it's a common symptom complex that we see in other conditions that are progressive as well. And so I think the challenge is going to be what's the best way to treat it or to predict the episodes and how to manage them. That may be more condition specific.
Another question on that. We'll come back to, I can see there's another question on CLN1 and John has his hand up.
Can I just talk a little bit about that? We're investigating the autonomic nervous system in our mice. We are looking at the heart and heart rate in the mice and looking to get some solid data as to its involvement. We know that many of the bits of the brainstem that are involved in autonomic function are clearly affected. So it's something we're working on. We work very closely with John Ostergaard, and we always try and look for correlates in mice, bearing in mind mice are not people, for things that we can measure and look for function and then see if our therapies actually treat them. And I showed data at the conference about brainstem pathology and the fact that we could actually treat that with enzyme replacement in CLN1.
That's great, John. It's really great to know that that's ongoing. Is that across multiple subtypes in your mice?
Yep. So we're looking predominantly in 1, 2, and 3, and we're looking at all that brainstem because it's very important for that interface between the brain and the rest of the body. We've obviously spent a lot of time looking at the peripheral nervous system, the enteric nervous system. The autonomic nervous system is on our hit list as well. It's just a question of how you do that and finding the right experts to work with. But fortunately, we have those in-house now.
And potentially sheep models too would be interesting to look at.
Yes, I wonder how to do it in a sheep, but I'm sure I can find someone who knows. Yeah.
We do have one other question around other therapies in CLN1 in development, but while we're just quickly on the parasympathetic dysautonomia, we thought this might be a popular topic today. But somebody's asking, Amber, our president, is asking, is it possible that this storming can be mistaken for seizures, dystonia, and hallucinations?
It's really hard, I would say, in the moment at home when these events happen. I mean, typically, the person may just appear very frightened and anxious. And of course, they're probably not going to be, hooked up at that moment to an EEG or anything else that could help us to know precisely whether there's any seizure activity happening. But I'd say it doesn't, it seems more a function of dysautonomia, dystonia, dystonic posturing can happen too. I would agree with you, Amber. There's lots of potential symptoms that could all kind of look the same, but I think it's just important to note that just on a kind of a strictly electrophysiological level, that tachycardia preceding then the other vital sign changes, it seems to be demonstrable and maybe a target that we could, a group of symptoms that we could help to alleviate.
If I can add to that, I spent time recently at the Nordic NCL meeting and sat down and discussed with the people who did this study, and they have good data that says that these events are taking place, but they're not at the same time as seizures, and they're not preceding seizures in those particular children. It was a question they were asked at the conference. They've looked very carefully back through their data, and it's not associated with the seizure. It's purely an autonomic dysregulation that they're seeing. So, and that's in a situation where they specifically measured to look to see if that was happening.
Yeah, that's right. And I think that's often, I've talked to the Rochester group about this as well, and John Ostergaard and the Danish group, and this is the so-called functional seizures, in that they present similarly, and I'm possibly talking out of turn because there's a real MD doctor on the call here in Courtney, but this is where they're talking about it can look like one, but if you were to do an EEG at the time, there'd be no true epileptic form seizure happening.
Yeah, I mean, I would just also mention that CLN3 patients at that stage of their illness are also very high risk for seizures as well. I mean,
Yeah.
we will also have an epilepsy diagnosis at that point. I just think for any individual spell, you know, it might be hard to know as a parent at home what exactly is going on. But I think from the literature, what we can say is it's just being recognized that apart from seizures, there are also these episodes of high sympathetic tone, sort of unmatched with the parasympathetic balance, and that seems to be creating these distressing events that are not seizures. but for sure, these are patients who are also at high seizure risk.
Yeah, complex. I know Dave has to jump off right on 10. There are a few more questions, and if any other panelists are happy to stay on, we can work through those. Dave, we don't think you're rude if you need to jump off to your next call.
My apologies, I do have to jump off. I'll try and listen in on my phone while I'm on the other thing, because I'm a multitasker, we all know that. So.
Excellent. Well, thank you so much for being here. We will continue on with the questions if everyone's happy to stay on. I know Amy has jumped on as well during the presentation. Amy, we explained that you were traveling to Washington and weren't able to get in there on time. She is here. Okay, so to the question around other therapies, for CLN1, John, I might let you talk a little bit about the ERTs in development and others.
Yeah, so I talked about a little bit about that earlier. And for CLN1, we're looking at a variety of approaches. First of all, is trying to do something about the brain itself. And there's two obvious strategies. One would be gene therapy, one is enzyme replacement. So we have done work where we're trying to develop an enzyme replacement approach for CLN1 disease, which is effectively, can we make Brineura, but for CLN1? So it's a different enzyme. You have to be able to make that enzyme and check that it has the right properties. We did that with Sandy Hoffman, who cloned the gene in the first place and made the mouse that we use. And we delivered that enzyme to the brains of our mice and figured out the best route to give it. Was it into the lateral ventricle, into the brain? Was it through a lumbar puncture? We figured out which was the best way, which turned out to be sticking it into the lateral ventricle. We showed that this had positive effects in mice, which was great. We think we can do better, and we're working on ways to do that. And then asked the question whether we could scale that up from treating a very simple mouse brain, which is about the size of my thumbnail, to whether we could treat a larger, more complicated brain, which was the sheep that we made, that Tom Wishart made in Scotland. And we showed that, yes, indeed, if we scaled the dose up, we could also treat those sheep. Now, some of that work was done together with a company called Collaboration Pharmaceuticals, who are pushing forwards to try and make more enzyme and to test whether that do the safety studies to see if that causes any particular problems or not, which they have to do. We are continuing on a slightly different track, which is to try and improve the efficacy of the approach we've got already by titrating the dose a bit better and changing the properties of the enzyme that we're using to see if we can do even better. The other approach is the gene therapy approach. That's one that we've obviously been applying to the brain and have been doing for many years, much of it together with Mark Sands here at WashU, and showed that you have to treat both the brain and the spinal cord in a mouse. So just treating one is not good enough. You have to treat both. And now we are scaling that up. As I say, we have federal funding to do those studies in CLN1 sheep, which are ongoing. The exciting news is that our first sheep were treated in the summer. We're now studying them and seeing how their disease progresses. And we'll be treating more in slightly different ways in the years to come. And hopefully we'll be able to report what is happening. And as I said, we heard at the conference about Spark Therapeutics, another gene therapy company who are also interested in using their particular virus and testing it in the sheep, and have been doing some of the groundwork towards doing that in the first place. Then on top of all of that is the work we're doing using gene therapy to treat disease outside the brain. So that's within the peripheral nervous system and the autonomic nervous system and the enteric nervous system, so in the bowel, And we've talked about both the peripheral and the enteric work before. The autonomic work is in progress.
Thanks, John. Just moving through some of the questions. We've got a question about the phase 3 child. How are the patients recruited for the child? Doctor referral only. So there's as long as you're in touch with one of the organizations, your physician, and they're aware that the trial is going on, even if you need to tell them that that trial is going on, you'll be able to connect with the trial sites. There is enough information out there. But if you're coming unstuck and you really need to connect through and have the assurance that you're connected to the right site, please do reach out to myself or Courtney or Amy or anyone from our organisations that can assist you. There was a question: could we be seeing these storming during the seizures during sleep? Yeah.
Sorry, I just responded by typing to a few of the questions.
Oh, sorry.
The answers
yes,
are visible, but to that question, I think yes, absolutely, it's possible.
Yeah. Yep. Sorry, I should look in the app.
Something even such as like a bad dream could be a provocation. I think it's hard to know, but there may not need to be any real provocation. I think these could occur awake or asleep.
Okay, great. Okay, a question about the going outside the CNS to peripheral nerves and the bowel, Batten disease outside the brain. What are the next steps for this study, John?
Our next steps are getting those studies published. We've done all this work. We're busy trying to get that. This is the first time anyone's ever treated the enteric nervous system by gene therapy. So we're trying to get that published in a fancy journal. And when you do that, you end up doing a lot more experiments to make the referees happy. But we hope that that story will be out in the open. They asked us to do a bunch more experiments, which are very sensible. We're trying to understand a lot of the biology of what's happening. But then the key question is, how do you do the same thing in a person? And that is what we are working on. We believe we need to understand the disease a bit better to do it, but at least we have proof of principle that there is something we can do about it. So that's what we're working on. We're working not just in mice, but we're actually working now looking at working with real gastroenterologists who work on bowel dysmotility. And we welcomed one of those to one of the meetings we had recently, a guy called Bob Hoykeroth, who's at CHOP in Philadelphia, who is one of the world's experts on this and how to treat bowel diseases. So the answer is we're calling in the experts to try and find ways that you could actually do this in people, because it's great. We can treat a mouse. It's wonderful. That's not what we need to do here. What we need to do is to treat children. So we are, as I say, calling in people whose everyday job is looking after these children and treating these children, and tag teaming with them to pick their expertise on how we're actually going to do this for real in people. So we will be doing that. We are doing that. We're looking to do more and more studies on how to do that. And we're doing exactly the same thing for the peripheral nerves as well. And that's turning out to be an interesting story that showed us we weren't as smart as we thought in the first place, that probably the things we're seeing out in the peripheral nervous system aren't really as a result of what's happening in the brain and the spinal cord, but they might actually be starting out in the periphery and may even have an origin in the muscles. So we're very interested in exploring that and trying to understand that. And we're doing that across multiple forms of NCL, CLN1, CLN2, and CLN3. And we see things happening. They happen differently, as you might imagine, because these diseases are not the same as each other. And we're doing a lot of work to understand it better, because if we understand it better, then we can actually treat it. And the good news is, yes, those things that happen in the muscle do also appear to respond to gene therapy. So we need to work on that. We need to understand it, and then work with the human experts who will help us to try and get those treatments into people.
Yeah, there's one more question there that I think is a really good kind of general question to finish off as well, and applicable across all the subtypes and all the studies that we've talked about today. But in terms of, you know, if we've seen efficacy for these gene therapies and other treatments in the animal models, say in sheep, you've gone into the large animal models, like we've seen with Nadia Mitchell and Dave Palmer's work in CLN5, how quickly, or what is the time frame? And it's a very big question, but what is the time frame once you've seen that efficacy for moving it into humans and getting, you know, these kids into trials?
Well, that's like saying how long's a piece of string, unfortunately.
A lot of
The
variance.
answer is not fast enough for any of us, neither as scientists nor as families. It all depends as we discover which company you end up tag teaming with. Now, it's a necessity and a fact of life that you can do a lot of good, valuable work federally or with foundation funding, that you can get a lot of preclinical work. But to actually take it into people, this requires a level of funding which is much higher. And that's where you hear the names of companies, and many of those companies are involved in doing that work. Some of them help us do that preclinical work. It's a question of partnering with people at the right time who have the right resources, who are able to push that forwards, which is what's happened for the CLN5 work that was done with sheep. We try to work with companies who will do that and move that forwards. As we discovered recently, some roadblocks happened that unfortunately stopped that progress. But in terms of timing, it depends who you're partnered with and how deep their pockets are and how committed they are to drive that forwards. And sometimes it does not matter how committed that company is, circumstances stop them from being able to do it. And that's frustrating for everyone. It's frustrating for people involved in the work. It's far worse, I know, for families. And we really wish we were in a situation where we could shorten that time frame. But we are at the mercy largely of funding and the people who are involved that we do this with.
Yeah, that's great. Thank you, John. Well, really, we're just down to some really lovely comments and thank yous from participants today. Thank you all for coming along out of your, you know, in your busy days and busy schedules to be here. I'd also like to note we will send out a survey after this. So if there are any other questions that weren't answered, please feel free to fill out that feedback survey and add your questions, and we'll get back to you as soon as we can. I want to say a very special thank you to all of our panelists today. Again, you know, such busy people and many fingers in many pies, doing lots of great work around the world, and with our Batten community first and, you know, foremost in your site. So thank you so much for being here, and we look forward to connecting with you again very soon. So I shall end the meeting here. Thanks all. Bye now.